I'm Dr. S.M.H, a PhD specialist in cellular and molecular biology, and your guide through the fascinating world of cellular machinery. Today, I'll be diving into 5 key aspects of the cell that will blow your mind and deepen your understanding of how life operates at the microscopic level. Before we jump in, I invite you to subscribe to my YouTube channel, Dr. S.M.H, where I explore the latest in scientific research and breakthroughs. Don't miss out on cutting-edge insights. Hit that subscribe button. Overview of the series. In this series, I'll be teaching you about 1. The nucleus and the endoplasmic reticulum, 2. The Golgi apparatus and lysosomes, 3. Mitochondria and the cytoskeleton, 4. Ribosomes and proteasomes, 5. Stress responses and viral invasion. Let's dive in and explore each one. Part 1. The nucleus and the endoplasmic reticulum. The nucleus. The cell's command center. Chromatin, euchromatin, active, versus heterochromatin, silent. Nuclear bodies, call, PML, and nuclear speckles. Endoplasmic reticulum, protein and lipid factory, rough ER, protein synthesis and folding, smooth ER, lipid metabolism and detoxification. Nucleus and ER, a vibrant and detailed illustration of the cell nu. Part 2. The Golgi Apparatus and Lysosomes. Golgi Apparatus, the cell's shipping hub, vesicular transport, clathrin, coping, copii-coated vesicles. Lysosomes and peroxisomes, recycling and detox units. Lysosomes degrade waste via autophagy endocytosis. Peroxisomes detoxify or ROS, synthesize lipids. Lysosomes and ProteasomesThe, vibrant illustration of lysosomes and peroxisome.pm. Part 3. Mitochondria and the cytoskeleton. Mitochondria, the cell's powerhouse, fission fusion, maintain quality control. Diseases, Parkinson's, Alzheimer's. Cytoskeleton, structural framework and highways, microtubules, actin filaments, and intermediate filaments. Mitochondria, a detailed illustration of mitochondria, showing tiatpain. Part 4, Ribosomes and Proteasomes. Ribosomes, protein synthesis machines, initiation, elongation, and termination phases. Proteasomes, protein quality control, ubiquitin proteasome system, ligases and deubiquitinases. Ribosomes and proteasomes, and a detailed illustration of ribosomes and proteasome. Part 5, Stress responses and viral invasion. UPR, unfolded protein response, IRE1, PERK, and ATF6. SGs, stress granules, aggregate untranslated mRNAs. ASFV invasion, hijacking protein synthesis via EIF2I. UPR and stress granules, /data, an illustration of the unfolded protein response.pn. Final call for in-depth explorations, and to stay ahead in science, subscribe to my YouTube channel, Dr. S.M.H. Let's explore the mysteries of life together. Part 1. The nucleus and the endoplasmic reticulum. The nucleus, the cell's command center. Scene setup. The camera zooms into the nucleus, revealing a vast expanse resembling a royal palace guarded by double-layered security gates known as the nuclear envelope. We enter through the guarded nuclear pores and step into the command center, where chromatin winds itself neatly like ancient scrolls holding the secrets of life. The nucleus is the cell's command center, managing all genetic operations. Here, DNA resides as chromatin, occasionally condensing into distinct chromosomes when the cell prepares to divide. In the heart of this palace lies the nucleolus, a bustling workshop crafting ribosomes, the artisans that will venture out into the cell to build proteins. Chromatin organization. Close-up shots reveal chromatin's dual personalities. Euchromatin, active, loosely packed, transcribing essential genetic codes. Heterochromatin, silent, tightly packed, storing ancient wisdom. Histone modification and epigenetics. Histones act as chromatin's librarians, organizing DNA with their protein spools. Post-translational modifications like methylation and acetylation dictate which genetic instructions will be read or archived, shaping the epigenetic code. Nuclear bodies. The camera pans to various corners of the nucleus, revealing specialized subdomains like Cajal bodies, PML bodies, and nuclear speckles. Cajal bodies, snRNP biogenesis. PML bodies, transcription regulation and DNA repair. Nuclear speckles, storage sites for pre-mRNA splicing factors. Nuclear transport. With a smooth dolly shot, the camera moves toward the nuclear pore complexes, NPCs, where molecular guards scrutinize every entrant and exit. Importins and exportins. Importins bring proteins with nuclear localization signals, NLS, inside. Exportins export proteins or RNA marked with nuclear export signals, viral hijacking of nuclear machinery. But every fortress has its weaknesses. Viruses like herpes simplex virus, HSV, and human papillomavirus HPV are cunning infiltrators. HSV1 ICP0 protein disrupts PML bodies, inhibiting the host antiviral response. HPV E6 and E7 proteins silence the guards P53 and RB, allowing viral genomes to replicate unchecked. The endoplasmic reticulum, protein and lipid factory. Leaving the nucleus, we enter a sprawling labyrinth, the endoplasmic reticulum, ER. Split into two wings, the ER hosts ribosome-studded assembly lines and serene lipid workshops. Rough ER and protein synthesis. The rough ER is covered in ribosomes, manufacturing proteins destined for secretion or internal use. Nascent polypeptides enter the ER lumen, where chaperones ensure proper folding. Protein folding and chaperones, ER chaperones like BiP and calnexin assist in folding, while misfolded proteins are marked for degradation. N-linked glycosylation, the first coat of paint on new proteins. Smooth ER and lipid synthesis. The smooth ER, SCA, is the cell's lipid brewery and detoxification hub. Lipid metabolism synthesizes phospholipids, cholesterol, and steroid hormones. Detoxification, like a water filtration system, the cytochrome P450 enzymes detoxify drugs and carcinogens. Calcium storage stores calcium ions for quick cellular signaling. The unfolded protein response, UPR, when misfolded proteins pile up, the ER sounds the alarm via the unfolded protein response, UPR. Inositol-requiring enzyme 1, IR1, splices XBP1 mRNA, producing an active transcription factor to upregulate protein degradation. Protein kinase like ER-resident kinase, PERK, phosphorylates EIF2A, reducing global translation. Activating transcription factor 6, ATF6, migrates to the Golgi and is cleaved into an active transcription factor, upregulating chaperones. Viral manipulation of the ER. Viruses manipulate the ER and UPR to their advantage. HCV in the UPR. Hepatitis C virus, HCV, activates the UPR but suppresses EIF2A phosphorylation to maintain viral protein synthesis. Dengue virus and ER remodeling. Dengue virus remodels the ER into replication vesicles. Let me know if you'd like any adjustments or if you'd like to move forward with Part 2. Number Part 2, the Golgi Apparatus and Lysosomes. The Golgi Apparatus, the cell's shipping hub. The camera sweeps across a stack of membranous sacs. This is the Golgi Apparatus, the cell's shipping hub. Protein modification and sorting. Proteins arriving from the ER undergo modifications in the Golgi. Glycosylation. Complex N-linked glycans and O-linked glycans adorn proteins. Proteolytic processing. Prohormones are cleaved into mature hormones. Lipidation and sulfation. Proteins receive lipid anchors and sulfate groups. Vesicular transport. The Golgi sorts cargo into vesicles for delivery to their final destinations. Clathrin-coated vesicles, head for endosomes and lysosomes. Copy-coated vesicles, retrograde transport from Golgi to ER. Copy-coated vesicles, forward transport from ER to Golgi. Golgi remodeling during viral infection. Viruses hijack the Golgi for maturation and exit. Coronaviruses assemble at the Golgi intermediate compartment, ERGC, and exit through the Golgi. Poxviruses convert the Golgi into viral factories. Lysosomes and peroxisomes, the recycling and detox units. Scene setup. We delve deeper into the cell and come upon two key units, lysosomes and peroxisomes. Lysosomes, cellular recycling centers. Lysosomes are the cell's recycling centers, packed with hydrolytic enzymes that degrade cellular waste. Autophagy. Damaged organelles and protein aggregates are recycled via autophagy. Endocytosis. Extracellular material delivered to lysosomes through endocytosis. Lysosomal storage diseases. Defective lysosomal enzymes lead to lysosomal storage diseases like Tay-Sachs. Peroxisomes, detoxifying organelles. Peroxisomes are the cell's detox units, neutralizing toxic substances and synthesizing lipids. Reactive oxygen species detoxification. Peroxisomes contain catalase to break down hydrogen peroxide. Beta-oxidation of fatty acids breaks down long-chain fatty acids into acetyl-CoA. Plasmalogen synthesis, vital for neuronal myelin sheaths. Let me know if you'd like any changes or if you'd like to continue with Part 3. Part 3, Mitochondria and the Cytoskeleton. Mitochondria, powerhouses of the cell. Scene setup. The camera moves through the cytoplasm and finds mitochondria, the cell's powerhouses. Energy production. Mitochondria generate ATP via oxidative phosphorylation. Electron transport chain. The electron transport chain pumps protons to create a gradient that drives ATP synthesis. Citric acid cycle. The citric acid cycle produces NADH and FADH2 for the electron transport chain. Mitochondrial DNA. Mitochondria possess their own circular DNA, hinting at their evolutionary origin as ancient symbiotic bacteria. Mitochondrial inheritance. Maternal inheritance ensures mitochondria are passed down through generations. Mitochondrial dynamics. Dynamic fission and fusion events ensure mitochondrial quality control. Mitochondrial fission splits mitochondria to remove damaged sections. Mitochondrial fusion combines healthy mitochondrial sections to form new functional organelles. Mitochondrial dysfunction. When the balance of fission and fusion is disrupted, mitochondrial dysfunction can lead to various diseases, including neurodegenerative disorders like Parkinson's and Alzheimer's disease. The cytoskeleton. Structural framework and highways. Scene setup. The camera pulls back to reveal the cell's internal scaffolding and transport highways, the cytoskeleton. Components of the cytoskeleton. The cytoskeleton comprises three main filament systems. Microtubules form highways for vesicular transport, compose the mitotic spindle. Actin filaments support cell shape and motility, facilitate muscle contraction. Intermediate filaments provide structural stability. Molecular motors. Molecular motors like kinesin, dynein, and myosin transport cargo along the cytoskeletal highways. Kinesin and dynein. Kinesin moves cargo toward the cell periphery. Dynein transports cargo toward the nucleus. Myosin drives muscle contraction and cell motility. Cytoskeletal dynamics and cell division. The camera zooms in on a dividing cell, showing the mitotic spindle orchestrating chromosome segregation. Mitotic spindle. Microtubules form the spindle that aligns chromosomes during metaphase, separates sister chromatids during anaphase. Viral manipulation of the cytoskeleton. Viruses often hijack the cytoskeleton to facilitate their transport and replication. Vaccinia virus and actin filaments. Vaccinia virus activates actin polymerization, propelling itself through the cell. HSV and dynein herpes simplex virus uses dynein to travel retrogradely along microtubules to reach the nucleus. Let me know if you'd like any modifications or if you want to move forward with Part 4. Part 4. Ribosomes and Proteasomes. Ribosomes, protein synthesis machine scene setup. We move back into the cytoplasm, where ribosomes, the cell's protein factories, are hard at work. Ribosome structure. Ribosomes consist of a large and a small subunit, each composed of RNA and protein. Translation initiation. mRNA, tRNA, and ribosomal subunits assemble into a functional ribosome. Elongation and termination. Amino acids are sequentially added to the growing polypeptide chain. Ribosome types. Free ribosomes synthesize cytoplasmic and nuclear proteins. ER-bound ribosomes synthesize membrane and secretory proteins. Protein synthesis process. We zoom into a ribosome translating a strand of mRNA, capturing the three phases of translation. 1. Initiation. The small ribosomal subunit binds to the mRNA, followed by the recruitment of the large subunit to form a complete ribosome. 2. Elongation. Amino acids are added one by one to the growing polypeptide chain as the ribosome moves along the mRNA. 3. Termination. When a stop codon is reached, translation halts and the ribosome releases the completed protein. Ribosomal regulation and antibiotics. Ribosomal activity is regulated by translation factors and environmental cues. Many antibiotics target bacterial ribosomes to inhibit protein synthesis. Viral hijacking of ribosomes. Viruses manipulate ribosomes to prioritize the synthesis of viral proteins. Picornaviruses. Picornaviruses like poliovirus cleave host translation factors, redirecting ribosomes to viral mRNAs. Influenza virus. Influenza virus hijacks ribosomes by encoding a cap-snatching endonuclease that cuts caps from host mRNAs and attaches them to viral transcripts. The proteasome, protein quality control. Scene setup. We zoom into the proteasome, the cell's protein quality control unit. Proteasome structure. Proteasomes are barrel-shaped complexes consisting of a 20S core and two 19S regulatory caps. Ubiquitin proteasome system. Proteins tagged with ubiquitin are recognized and degraded by the proteasome. Ubiquitin ligases attach ubiquitin to target proteins. Deubiquitinases remove ubiquitin tags, regulating protein fate. Protein degradation process. The camera follows a ubiquitinated protein as it is processed through the proteasome. 1. Recognition and unfolding. The 19S regulatory cap recognizes the ubiquitin tag and unfolds the protein. 2. Translocation and degradation. The protein is translocated into the 20S core, where it is degraded into small peptides. Proteasomal regulation. Proteasomal activity is regulated by ubiquitin ligases, deubiquitinases, and cellular signaling pathways. Proteasome inhibition and therapy. Proteasome inhibitors like bortezomib are used to treat cancers like multiple myeloma by inducing the accumulation of misfolded proteins, leading to cancer cell death. Viral manipulation of proteasomes. Viruses manipulate the proteasome to evade immune detection. Epstein-Barr virus, EBV. EBV encodes a deubiquitinase that removes ubiquitin from viral proteins, preventing degradation. HIV. HIV protein VIF recruits ubiquitin ligase to target ApoBec3G, a host antiviral enzyme, for degradation. Let me know if you'd like adjustments or if you'd like to proceed with Part 5. Part 5. Stress responses and viral invasion. Stress responses, managing cellular adversity, setup. We explore the cell's stress responses that kick in when adversity strikes. The unfolded protein response, UPR. The UPR aims to restore ER homeostasis by upregulating chaperones and reducing protein synthesis. PERK pathway phosphorylates EIF2A, reducing global translation, promotes the translation of UPR target genes like ATF4. IO1 pathway splices XBP1 mRNA, producing an active transcription factor, XBP1s, that upregulates protein degradation. ATF6 pathway. ATF6 translocates to the Golgi, cleaved into an transcription factor that upregulates ER chaperones. Stress granules, SGs, are aggregates of stalled translation complexes that form during stress. Composition. SGs contain untranslated mRNAs, ribosomal subunits, and RNA-binding proteins like G3BP1 and TIA1. Role in viral infections. SGs often limit viral replication by sequestering viral mRNAs. However, some viruses inhibit SG formation to maintain viral protein synthesis. Heat shock response, HSR. The HSR is activated in response to elevated temperatures and other stresses. Heat shock proteins, HSPs. Molecular chaperones like HSP70 and HSP90 assist in protein folding and prevent aggregation. HSF1 activation. Heat shock factor 1, HSF1, trimerizes and translocates to the nucleus, where it upregulates HSP expression. The ASFV invasion, how viruses hijack cellular machinery. Scene setup. We transition to the final chapter, where the African swine fever virus, ASFV, plots its invasion. ASFV hijacks protein synthesis. ASFV antagonizes protein synthesis shutdown via EIF2A dephosphorylation. EIF2A dephosphorylation. ASFV ensures efficient viral protein synthesis by dephosphorylating EIF2A, preventing the formation of SGs. Salubrinol as a countermeasure. Salubrinol, an EIF2A dephosphorylation inhibitor, counteracts ASFV replication. ASFV subverts the host's stress responses. ASFV manipulates the UPR and inhibits SG formation. ER stress activation. ASFV activates ER stress but suppresses PERK-mediated translation inhibition. Inhibition of SG formation. ASFV protease PS273R cleaves G3BP1, preventing SG formation. Case study. The ASFV GZ201801-2 strain. We delve into a case study of the ASFV GZ201801-2 strain, revealing how it manipulates host cellular machinery for efficient replication. ER stress and UPR activation. The GZ201801-2 strain activates all three UPR branches, enhancing protein degradation while maintaining viral protein synthesis. Inhibition of EIF2A phosphorylation. The strain's powerful dephosphorylation of EIF2A ensures the continuation of viral protein synthesis. Counteracting SG formation. ASFV-mediated EIF2 dephosphorylation prevents sodium arsenide-induced SG formation. Potential therapeutic approaches. Salubrinol. Partial inhibition of ASFV replication by counteracting EIF2A dephosphorylation. Targeting UPR pathways. Inhibition of specific UPR pathways may reduce viral replication. Conclusion. The camera pans out, revealing the entire bustling cellular city. Understanding cellular machinery and how viruses hijack it offers invaluable insights into disease mechanisms and therapeutic targets. This journey into the microscopic world of the cell highlights the beauty, complexity, and fragility of life at the molecular level. Subscribe to our channel for more scientific explorations.